This recording is the somatic nervous system, part 1 of 4. Sensory perception helps us understand what's going on in our environment, both internally and externally. The stimulus causes sensory cells to have an action potential that is going to be relayed to the central nervous system. There, the stimulus is going to be integrated with other sensory information and higher cognitive functioning in order for the stimulus to become a conscious perception. Awareness is just the stimulus being sent to the primary somatosensory area of the brain. Perception is how we perceive the stimulus. I'll give you an example of this. If a person hears a loud bang, well, that's awareness of sound. If the person compares that to other sounds, then the individual might perceive that as the sound of a firework going off. And that's where perception comes into play. Sometimes sensory perception leads to motor responses, sometimes it does not. Another example of sensation versus perception is in this picture where we see the sensation is the scent of the rose. However, the perception is that the scent is that of a rose rather than that of a banana. There are different types of sensory receptors, and these can also be classified according to the structure of the receptors. For instance, we know that there are some receptors that simply are arranged as free nerve endings. In this sensory neuron, the dendrites act as the free nerve endings. You'll notice this is a unipolar neuron. Remember that the cell bodies of unipolar neurons are found in ganglia in the peripheral nervous system. Sometimes we'll see a sensory receptor that is encapsulated, such as is the case where the dendrites are in a special encapsulation. We see this in Pacinian corpuscles, as well as Meissner's corpuscles. Sometimes receptor cells can be specialized, and here we have a specialized receptor cell in the form of a rod. A rod is a specialized photoreceptor cell. There are also photoreceptor cells called cones, which have a very specific shape and structure so that they can interpret different types of stimulus. We can also look at receptors based on location. Exteroreceptors, also known as exteroceptors, tend to be located close to the external portions of our body, such as our skin, to give us information about our external environment. And we've already seen free nerve endings in the epidermis. We've seen Meissner's corpuscles in the papillary layer of the dermis, Pacinian corpuscles located in the reticular layer of the dermis, and there are also Krauss and bulbs and Ruffini endings found in the dermis, as well as hair follicle receptors. So these are all different exteroreceptors. They give us information about our external environment. Interoreceptors or interoceptors can give us information from our internal organs and tissues, and they have sensors that will sense things like a change in blood pressure, which we know are baroreceptors, or a change in chemical concentration that we see in chemoreceptors. You'll remember from the previous chapter that The carotid bodies have chemoreceptors for oxygen and carbon dioxide, and the carotid sinus has baroreceptors to detect changes in blood pressure. And then we have proprioceptors. Proprioceptors are usually located near moving body parts, muscles, and joints to help interpret the position of the body in space. Receptors can also be based on function, categorized on the basis of the type of stimuli they transduce or convert into an action potential. For instance, mechanoreceptors are receptors that will respond to changes in physical distortion. Our mechanoreceptors include those that detect changes in blood pressure as well as touch and physical pressure and vibration. We also have thermoreceptors that detect changes in temperature. Nociceptors detect cell damage and transmit signals that there is damage, and pain will be the result of nociceptors being stimulated. Chemoreceptors are important for detecting changes in chemicals, and photoreceptors are important for detecting light. Osmoreceptors will respond to changes in solute concentration, osmolarity. When we look at general sensory receptors, these are important because they provide our central nervous system with information about what's going on around us as well as inside us. So we have information that's internal and external. Our general senses are those that help give us sensitivity to things like touch, temperature, pain, pressure, vibration, as well as proprioception. Remember, proprioception is that information about where your body is in space. Obviously, someone who's a gymnast, which you can see in the picture above, is going to have very finely adapted proprioceptors because they really have to know their body position, as do divers. When a diver goes off a diving board, that person has to know how many rotations or twists or turns he or she has gone through to unfold at just the right moment to hit the water in a particular form. The interpreted intensity of the incoming sensation is due to the frequency of arriving action potentials. Remember, action potentials all have the same amplitude, but it's the frequency that determines the intensity of the stimulus. Our general sensory receptors are actually not evenly distributed through our body. They're distributed unevenly. And the information that we get from these receptors is what we refer to as sensation. However, the awareness of what the sensation is and how we interpret it is our perception. When we look at somatosensation or touch, this is a general sense, and touch is really just one of the types of somatosensations. It's the most common type with which we are familiar, but it also includes things like pressure, vibration, tickle. We have itch, pain, proprioception, kinesthesia. These are all different somatosensations. The receptors for somatosensations are spread throughout the body. Again, not equally, but they are spread throughout the body. Many are located in the skin, and some are found in muscles, tendons, joint capsules, ligaments, as well as in the walls of visceral organs. This takes us to the mechanoreceptors of hairy skin. We already know about the hair follicle receptor, which detects movement when a hair is moved, if there's something crawling on the skin, on the surface. Pacini and corpuscles for deep pressure and vibration. Ruffini endings or Ruffini organs, which are involved in detecting stretch. Merkel's discs are kind of neat. They have a high tactile acuity for an object's physical features like shape, texture, and edges. And the whisker follicles of many mammals have a lot of Merkel's discs, which we know are Merkel's cells or tactile cells. So they're for light touch. And then our free nerve endings that extend up into the epidermis, which this picture does not show accurately, are for pain, temperature, and mechanical deformation, as well as nociception. Other mechanoreceptors include muscle spindle fibers, which are located in the muscle belly, and they consist of different fibers, including extrafusal fibers and intrafusal fibers, and they basically carry information to the central nervous system regarding the length of the muscle to have better coordination in terms of muscular contraction so that the body can balance accordingly. If a person were to walk across a log to cross a creek, there are different muscles that may start to contract due to the information that's being sent to the central nervous system to help coordinate balance. And again, muscle spindle fibers are found in muscles, and they provide information about the length of the muscle, whereas Golgi tendon organs are located in tendons, and they monitor the strength of, they monitor the stretch of the tension of a tendon, thereby monitoring the tension of the muscle that they attach to bone. And that's how they help prevent muscles and tendons from tearing when they are subjected to potentially damaging stretching forces. They inhibit agonists and activate antagonists. So muscle spindle fibers found in muscles, Golgi tendon organs found in tendons, and they both act to help protect the body in some way, shape, or form. Muscle spindle fibers help us by coordinating balance. Golgi tendon organs help us by preventing damage from excessive stretching forces. This will take us to special sensory receptors, which include olfaction, which is the process of smell, vision, which is the process of sight, gustation is tasting, equilibrium is balance, and hearing. The receptors of special sense organs are very structurally complex and are located in specific special sensory organs like the tongue, the nose, the inner ear, and the eye. And the information that's provided by these receptors is going to be sent to a specific area of the cerebral cortex as well as to the brainstem. Stimulus is going to be translated into an action potential. So even though we get light stimulus into our eyes, it has to be translated or transduced into an action potential in order for nerves to communicate with areas of the central nervous system. We're going to start our journey of special senses with gustation, which is taste. And taste buds are basically chemoreceptors that are found on the tongue and in certain areas of the pharynx, which is your throat. We have approximately 3,000 taste buds that end up declining as people get older. Taste buds are usually most commonly associated with specific structures, including the foliate papillae. They have that name because they are leaf-like appearing structures. The valate papillae and the fungiform papillae. You'll notice that these papillae have invaginations or infoldings where the taste buds will be found. So here we have an enlarged section of a valate papilla. And when we look more closely at the taste buds, you can see that there are tiny little gustatory hairs that act as the receptors for different types of chemicals. And there is an opening to the taste bud called a taste pore. This is all embedded in stratified squamous epithelium found on the surface of the tongue. And the taste bud is made up of gustatory epithelial cells, which you can see in dark purple, as well as basal epithelial cells, which help support the gustatory epithelial cells. And we can also see the taste fibers of the cranial nerve supplying this taste bud. Remember that cranial nerve 7 which is the facial nerve, not only supplies muscles for facial expression, it also has that sensory portion for the anterior 2/3 of the tongue. Here we have more pictures of taste buds, and you can see the different areas where these are found on the tongue. And again, most of the taste buds are going to be found in the infoldings of the papillae of the tongue. You'll remember from looking at the dermal papillae that a papilla is just a nipple-like projection. So again, we have the fungiform, foliate, and veillate papillae. Some books refer to the veillate papillae as circumvallate because they are circular structures. Each taste bud has approximately 50 to 100 cells, gustatory receptor cells, and these receptors have tiny little microvilli that are going to increase surface area, and these microvilli are bathed in saliva. And so these gustatory hairs basically receive the different types of chemicals, and then those chemical signals are converted or transduced to send action potentials along sensory neurons. Remember, anterior 2/3 of the tongue is the facial nerve, cranial nerve 7. The back third of the tongue is cranial nerve 9. That's glossopharyngeal. And the basal cells are basically stem cells in the taste buds that replace the gustatory cells and support them physically about every 7 to 10 days. So far we've talked about 2 cranial nerves involved in gustation. Cranial nerve 7, which is the facial nerve, innervates the taste buds for the anterior 2/3 of the tongue, and cranial nerve 9, which is glossopharyngeal, innervates the taste buds for the posterior third of the tongue. However, there is another cranial nerve involved, and this is cranial nerve 10, which has a branch that innervates the larynx. And once sensory information goes from these cranial nerves, those neurons will synapse with the solitary nucleus in the medulla oblongata. From there, a neuron will carry information to the thalamus, because the thalamus is the gateway for all incoming sensory information to the cortex, except for smell and proprioception. At the thalamus, the information will synapse with cell bodies in the ventral posteromedial nucleus of the thalamus, and then from there information gets carried to the gustatory cortex in the insula. Now please, I've mentioned very specific nuclei here for taste. However, what I really want you to know is that taste can be carried on cranial nerves 7, 9, and 10, which are facial, glossopharyngeal, and vagus. And then finally to the gustatory cortex found in the insula. Now, some sensory information has the ability to undergo adaptation where the brain no longer processes the information because it just gets used to it. And taste can actually completely adapt anywhere from 1 to 5 minutes. So it adapts pretty rapidly and completely. 80%, sometimes books say as much as 85% of our taste is due to what we smell. So without smell, our food ends up lacking a lot of flavor. The mouth also contains thermoreceptors, mechanoreceptors, and nociceptors by way of the trigeminal nerve, and this gives us information about texture and temperature of foods, which can either enhance or detract from their taste. Chili peppers will excite pain receptors, and some people that brings pleasurable effects. Some people like really, really spicy food and aren't happy unless they're sweating when they eat something spicy. Olfaction is the sense of smell. And the sense of smell, just like the sense of taste, is going to involve chemoreceptors. Remember, with taste buds, those taste buds have little gustatory hair cells which are bathed in saliva, and the chemicals in the foods that we eat are carried in the saliva to those receptors on the taste hairs of the gustatory cells. When we look at the olfactory sensory neurons, we see that these bipolar neurons have dendrites that extend down towards the layer of mucus found on the epithelial surface inside the nasal cavity. And they have branches on their surface that are cilia, and that helps to increase the surface area for chemoreceptors for these dendrites. In between the olfactory sensory neurons, we also have supporting epithelial cells, and basal cells are those stem cells that can divide and replace the olfactory sensory neurons. You'll notice that the axons of these bipolar neurons are going to go through the olfactory foramina of the cribriform plate of the ethmoid bone. The axons will synapse with other neurons in the olfactory bulb, and those axons will go along the olfactory tract to the olfactory cortex in the temporal lobe. You'll notice there are other axons that go from the olfactory tract to the hypothalamus and the limbic system, where they will become associated with emotion and memory. The olfactory nerve is cranial nerve 1. There are a lot of mnemonics that are available for remembering the cranial nerves in terms of, numbers 1 through 12. And there are also mnemonics for remembering which ones are sensory and which ones are motor. As far as the sensory and motor portion, you should be able to tell that the sense of smell is not motor. That's a sensation, as well as vision. So I'm not really concerned that you memorize. Some say Merry Money, that mnemonic is not really important to me. However, you really should have a good mnemonic that you'll remember for the cranial nerves in terms of which number they are and what that number represents. Years ago, when I had to learn this, I came up with my own mnemonic that made sense to me, because O, to touch and feel green veggies, meant nothing to me. O, that's 3 O's. Which O is what? So I developed my own mnemonic that made sense to my brain, and years later I still remember Ollie opted occult to try abducting facial vestibules, give a hoot. And that's how my brain remembers Ollie, olfactory, opted, optic, occult, oculomotor, 2 is trochlear, that was the closest thing I could come, trigeminal, abducting, abducens, facial, vestibules, vestibulocochlear, give, to me, is GV, glossopharyngeal, vagus, A, accessory, hoot, hypoglossal. So if you come up with your own mnemonics for certain things, you might find that helpful if you run across a mnemonic that just doesn't make sense, or you can't use because it just doesn't stick. Here we have a closer look at the olfactory epithelium. You can see the olfactory receptor cells. They are bipolar sensory cells, and you can see that the dendrites extend into the lumen or opening of the nasal cavity, where they're enclosed and embedded by mucus, and you can see that there are molecules that will carry the sense of odor to the extensions of the dendrites, which are technically called cilia on these particular cells. The basal cells are simply the stem cells that will undergo mitosis, replacing the neurons, the olfactory receptor cells, when they undergo apoptosis. We see the olfactory epithelium on the inferior side of the cribriform plate of the ethmoid bone, as well as the superior portion of the perpendicular plate of the ethmoid bone and the superior nasal conchae. This particular slide reminds us that olfactory pathways are very closely linked with memory, emotion, and taste. It also says that it's the only sensory information that directly reaches the cerebral cortex by bypassing the thalamus. Previously, you've heard me include proprioception as being one of those sensations that doesn't go directly through the thalamus prior to going to the cerebral cortex. And that is because proprioception actually usually goes through the cerebellum, which makes it very distinct from these other sensations. And I just wanted to point out that particular bit of information for you, because you may read that proprioception goes through the thalamus in your book. However, there's more involved with it. This takes us to some physiology. Olfactory neurons are replaced frequently. However, that replacement declines as people get older. Usually, olfactory neurons, these bipolar neurons, live only 1 to 2 months. The sense of smell is the most rapidly adapting sensations. It occurs quickly, so you can smell new smells rapidly. That's why if you ever walk into a fish market and you get this overwhelming sensation that there's fish nearby, that sense of smell will decrease so you don't notice it. And it is the most rapidly adapting sensation that we have. There is a very low threshold of stimulus required, and only small concentrations of molecules are needed to stimulate olfaction. Again, from our olfactory pathways, we've got the flow to the hypothalamus, the thalamus, and other regions of the limbic system that are going to be involved. It doesn't have to go to the thalamus before it gets to the cortex. It can bypass the thalamus to get to the cortex to be sensed. However, there are olfactory pathways that will take it to other regions as well. We get different types of emotional responses elicited, such as smelling smoke or gas and feeling that there's danger nearby, or a skunk. If you've ever been in a movie theater and smelled popcorn popping, that can stimulate your appetite, as well as the smell of bacon or onions. And then there are other odors that are very unpleasant, like sometimes people find the odor of pepper or paint fumes or rotten food or exhaust very unpleasant. This ends this slide presentation. We're going to move on to part 2.